Hi, my name is Florian. I'm a postdoc working with Oni Basu and Sebastian Pott at the University of Chicago. And I wanted to talk about how you can perform PCA on single-cell RNA-Seq data in 3 simple steps. And as you probably know, PCA is a key step in many single-cell RNA-Seq analysis workflows. So often when you're doing visualization with t-SNE or UMAP, or you're doing clustering or trajectory inference or many other things, what you're actually working with are these principal component scores, which are the result of a principal component analysis. So that's why I wanted to make this video, because it's a very important step. I was also inspired by this recent paper called Impact of Data Preprocessing on Integrative Matrix Factorization of Single-Cell Data, which got quite a bit of attention on Twitter. And I just have a quote here from the introduction. They write, "Matrix factorization methods, including principal component analysis, are central to single-cell RNA-seq data analysis pipelines, but are often treated as black boxes within computational pipelines, with little consideration of what steps are included." So I completely agree with that. And I think it's a missed opportunity when you're just learning about how to analyze single-cell RNA-seq data, when you're just sort of skipping over this step and never really take a look at what's happening there. Because, like I'm going to show you, I think it's actually, it doesn't have to be complicated, and you can better understand what happens to the data there. And in this paper, they, I think they did a great job sort of explaining a lot of things and giving definitions. And then they talked about different ways of doing PCA, but at the end, it wasn't clear to me, what the actual recommendation was for how to actually do a PCA. So I wanted to make a video about how I do PCA and how concretely you can get pretty good PCA results. So I'm going to talk about a little bit about why we're doing PCA, and then how to perform it in these 3 steps. And I'm going to show some examples, and at the end I'm going to add a little discussion. Okay, so why are we doing PCA? So I think there's 3 main reasons why we're doing PCA on single-cell RNA-seq data. So when you're just starting out with the raw, what we call the raw data, which is just the matrix containing all the UMI counts for all the cells and all the genes, if you focus on, let's say, protein coding genes, there's around 20,000 of them in human and mouse. And so what that means is that each cell, the expression profile of each cell is a vector with 20,000 entries. So we're saying that the data is 20,000 dimensional. But we also know that in reality, the dimensionality of the data is actually much lower. Why? Because many genes are actually co-expressed. So co-expression of genes happens all the time. They're highly correlated. And let's say, imagine like a toy example where you maybe have just four cell types in your data, and assume for a second they're all perfectly biologically homogeneous, which they may not be, but assume that they are for a second. If you have 4 cell types that are homogeneous, then really your data lives in 3 dimensions, no matter how many genes you're measuring. So this is kind of the picture. Let's imagine like each cell type is like this cloud, and maybe the reason why there's a cloud is because of technical noise. But you see all these 4 clusters, they all live in this 3-dimensional space. So we only need 3 dimensions to represent all the expression information in that matrix, even though you might measure 20,000 genes. So you could, of course, just say, well, okay, that's nice to know, but why not just ignore that? Why not just work with all the 20,000 genes? So that often causes problems in many situations. So there's this thing called the curse of dimensionality, which I feel like sometimes just used as kind of this catch-all phrase for bad things are going to happen from a statistical point of view. But more concretely, one of the things that happens in very high dimensions is that all the distances between the cells become very similar to each other, and that can be a problem. Also, when you're fitting a model, then you have to kind of deal with all the 20,000 variables in this model, which can also be a problem statistically. It can lead to overfitting. So very high dimensional data is often problematic from a statistical point of view, but it's also computationally inefficient, because now all of the operations you're doing with the data, first of all, you have to store this large matrix in memory all the time, and whatever you do to it, you have to store the result of that, which is also going to have 20,000 genes. So it's not very efficient. It's also slower. When you're fitting a model, it's often much slower to fit a model with 20,000 variables than with 20. So it's computationally inefficient. And the third reason, which I think is often not appreciated enough, is that PCA is actually also doing a lot of good work in terms of separating noise from true biological expression differences. So the reason for that is because PCA looks for correlation structure or covariance, and like we discussed, because of the co-expression, there's a lot of correlation structure typically in expression data, and the PCA is going to pick up on that, and so it's going to pick up on these biological expression differences very, very well. Whereas the technical noise, there's a lot of technical noise in single-cell RNA-seq data, but it doesn't have any correlation structure, so it's not correlated among genes. So so the PCA can't really catch that, and that's a good thing, because then that means that the PCA is going to separate true expression differences and technical noise. So this is a figure from one of my papers, and just to show this point a little bit more, so the idea of PCA is you're decomposing this expression matrix into two smaller matrices, or two different matrices at least. One contains the principal component scores and one the principal component coefficients. And the idea is, if you're just keeping the first couple of principal components, those components capture biological expression differences or biological heterogeneity, and the higher principal components, they only capture the technical noise. So it's a good way to separate expression differences and technical noise. So how do we actually do this now? So I'm going to first show an overview. So like I said, we're starting with this UMI count matrix. Typically what I do is I exclude mitochondrial genes, and I focus on protein coding genes, not so much on the tens of thousands of non-coding genes. But that's a separate kind of discussion, and it doesn't affect what you do here. Doesn't really affect the next steps. So independently of how you're pre-processing your matrix, the next steps should stay the same. So the first step is scaling, then you're transforming the data, and then you're doing PCA, and then you get your PC scores usually. And so I'm now going to talk about each step separately and go into detail of what that actually means, and how to actually do each step. So first, with the scaling, basically what we need is the median transcript count. By that, what I mean is you go over all the cells, and for each cell you sum up all the UMIs for all the genes, and that sum is what I typically call a transcript count. Sometimes it's called the library size. So let's call that TI, and I as the index for the cell. And then you just determine the median of all those TI values, and let's call that median C. So that's the median transcript count. And then you scale the UMI values for each cell by the factor C divided by TI. So that's a different factor for each cell, because each cell has a different transcript count here. And what happens is, so here's some real data where I've ranked all the cells by their transcript count or their UMI count. So some cells have, very high UMI count. Some cells have very low ones. Most of them have something in between. After the scaling procedure, so the median happens to be 5,783. After the scaling procedure, all the cells now have the same transcript count, right? And they're all equal to the median. So that's the idea behind scaling. It's pretty simple. So this step does two things. First of all, obviously, it means that now all the expression measurements are relative. They're relative to whatever, to that number C, or, so it's no different than basically working with percentages or concentrations. So they're basically now all concentrations, not absolute expression levels. But it also does this thing where it sort of removes something that's called efficiency noise. If you want to know more about efficiency noise, you can read this paper. The idea is that when you're doing the single-cell RNA-seq protocol, each cell gets encapsulated in some droplet, let's say, and the chemistry, you capture the mRNA from each cell, and for some cells that that for whatever reason that happens a little bit more efficiently than for others. For some cells you're capturing more of the actual mRNAs in the cell, and for other cells fewer. And we think that that's just technical differences, and sometimes randomly happens, depending on the chemistry and maybe the primers for each and the barcode of each cell. So by scaling, you're basically getting rid of most of that variation, and that's a good thing. So now we have one less source of noise in our data. Okay, so step 2 is the transformation step. That's kind of my favorite step because it's also very simple, and it does a lot of work, even though you may not realize it. But it really makes this whole approach work, this transformation, right? And the idea is just to replace all the values in the matrix Xij, where I is the cell index and J is the gene index. Just replace that with some value Yij, where Y equals f of x, and so f is some function you have to choose. And so it's the same function for all the values, so it's very simple. What I recommend for f is this transform called the Freeman-Tukey transform, which is square root of x plus square root of x plus 1. There's other choices you can make. The most popular one by far is the log transform, which is a natural log of x plus 1, where there's the pseudo count. That's what's called the pseudo count sometimes. And then there's also another square root transform called the Anscombe transform, which looks like this. Like I said, I would recommend this one. I think it's the most principled choice for theoretical reasons. But these other choices, in my experience, they also work. And typically, I think the differences aren't dramatic. Obviously, it is a difference. So, but basically, all of these choices should approximately allow you to get good results. So this step basically performs also two very important things. So first of all, it prevents the entire analysis in the PCA from being dominated by the highly expressed genes. So expression levels often span sort of 2 orders of magnitude or more. So obviously, if you didn't transform the data, then the highly expressed genes, in absolute terms, they just have a lot more variance. So if you're doing PCA, it looks for covariance. So it will be very biased towards these highly expressed genes because their absolute values are so much larger than all the other genes. So we don't really want that. We don't want, what's the point of measuring, all the genes if in the end the PCA is driven by 5 genes or something? That doesn't make any sense. So we want to squish the high expression values a little bit, and obviously the log compresses the high values a lot more than the square root, but both of them basically downweight the highly expressed genes, or make them smaller so that the slightly less highly expressed genes get a chance. The second thing it does is it has more to do with the technical noise that I mentioned, and so it's more, you have to kind of understand a little bit more the technical noise, the profile of the technical noise in single-cell RNA-seq data. But basically, especially the square root transforms, what they do is they weigh each measured expression level by the signal-to-noise ratio that we expect for that expression value. And so that's very good because basically it makes kind of a good use of our data. It means that genes that are measured, highly expressed genes that are measured much more accurately than lowly expressed genes, they get more weight, but not too much. I mean, because if you didn't do any transform, they would get too much weight. But they get more weight than the lowly expressed genes, and especially very lowly expressed genes, the technical noise in single-cell RNA-seq data for those is so high that there's basically almost no useful information in there. The signal-to-noise ratio for those genes is so low that we basically just want to ignore them. And so that's why a lot of other protocols, you remove some genes, but if you actually apply this transform, for example, it will make it so that the lowly expressed genes, they basically don't contribute at all anymore to the analysis, which is good because they don't contain any useful information. So these square root transforms are very proportional to the signal-to-noise ratio. This log transform is a little bit it downweights the highly expressed genes a little bit too much, so it gives them less weight than they should have based on their signal-to-noise ratio. But it still gives you, as in my experience, it still gives you useful results. It's not like catastrophic or anything. So the last step after you do the transformation is to do the PCA. PCA is very standard, so every language has packages or functions for it. Of course, you have to kind of figure out how many principal components you want to keep. So if you don't know, principal components are ordered by the amount of variance they explain. Typically, I think for single cell data, starting with 30 principal components is a good choice often. But I'm going to talk a lot about that a little bit more in the next slide. The other thing I think is important in this step is that you want to make sure we perform PCA as fast as possible. So depending on which implementation of PCA you're using, it can either take 5 minutes or 5 seconds. And that makes a difference because I often do this thing, this PCA is something I do all the time, and I don't want to wait 5 minutes every time I do that. I like it to take 5 seconds. So some background, if you're interested, otherwise you can just skip ahead. So let n be the number of cells and p the number of genes in your data. Then the total number of principal components that's present in the data, or that you can get, is the minimum of n minus 1 and p. So if a data set has 1,000 cells and 20,000 genes, there should be genes here, then the number of principal components is 999. So it doesn't have 20,000 principal components. It has 1,000 minus 1. But we're not interested in 999 principal components. We're only interested in maybe the first 30 or so. So there's an algorithm called randomized SVD, which allows you to determine the first couple of principal components very, very fast, much faster than if you wanted to determine all the principal components. So if you this algorithm is described in this paper here, and so you want to make sure you use that algorithm because that's much faster than everything else. So here's an example on actual PBMC data, 10,000 cells, I think. So the randomized SVD took 2.4 seconds. The full SVD, what you use to get all the principal components, took more than 5 minutes. So that's a difference of 138-fold. So we don't want to wait 138 times longer than we need to do our PCA. So you want to make sure you use the right implementation. So in Python, it's basically chosen automatically when you're using the scikit-learn PCA tool, but you can also specify it and say, I want to use the randomized algorithm. In R, there's a package called RSVD that implements this randomized SVD algorithm. So just make sure you're using one of those implementations and not just a standard SVD, because that's very slow. So onto the question of how many principal components should you use. So that's not an easy question to answer, but the good thing is you can kind of get away with approximating it fairly well in many cases. So my rule of thumb is that it's better to use too many than too few principal components. So what happens is when you use too many principal components, the additional principal components, they capture more of the technical noise, but not much more, because it's sort of, I think I've shown you, the increases very slowly, the heterogeneity. If you use too few principal components, then you're losing expression differences, which is obviously very bad. So this is not so bad, this is very bad. So it's better to include too many principal components. So for data sets that aren't too heterogeneous, that don't have too many cell types, I think 30 principal components is a reasonable choice. When you have a lot of cell types, I mean, I'm not talking hundreds, I'm talking maybe a few dozen or so. Let's say more than 20 cell types or something. Then maybe you should use maybe 50 principal components. So, and if you're not sure, just start with 50 principal components. So just to remind you, this is the separation between expression differences and technical noise. So this is what I was referring to earlier. So when you look at the cumulative variance explained for this PBMC data set, this example here, so you see that the first couple of principal components already explain 30% of the variance, and then it sort of starts to more slowly increase here, and it kind of keeps going at a pretty constant rate here. So somewhere in between here, there's this point where we're changing, where we're no longer capturing biological expression differences, and we're starting to only capture technical noise. And because that's not correlated, each principal component doesn't capture a lot of variance. So you see that's why it's increasing so slowly. But it's sort of not clear if you look at it, whether you should choose 20 or 30 or 40 or 50 principal components. It's kind of hard to tell by eye. But let's say you picked 50 instead of 30, then you're including an additional 1 or 2% of variance compared to more than 30% biological variance. So that's not horrible. That's just a small increase in technical noise. So that's why I think typically it's not going to result in any catastrophically bad result if you include more principal components. There's an approach called molecular cross-validation, or MCV, by Batson et al. If you use that, then you can sort of estimate the optimal number of principal components more systematically instead of just guessing. So in this case, for this data set, it's actually 30, according to this approach. And but the thing is, though, this is much more complicated. It takes much longer. So if you're interested in it, take a look. I included it in my in the software that I published recently, Monet, the Python package for analyzing single-cell RNA-Seq data. And you can also look at this paper. But it's like I said, you can approximate the number of principal components, and that usually works fine. So to summarize, you start with your UMI count matrix, then you scale to the median transcript count. Don't scale to like some ridiculously large number, like 1 million or something. That has very bad effects because that messes up the next step. So scale to the median or the mean, but I think because sometimes you have these cells that have a lot of transcripts, the median is safer because it's more robust to these outliers. Scale to the median transcript count, then do a transformation. I recommend this square root based transformation, but like I've shown you, alternatives are also possible. Then determine the first 30 to 50 principal components and use the sufficient implementation of randomized implementation that I mentioned. And that's it, and then you have your principal component scores, and that works, I think, very well most of the time. And it's a very simple, you can, if you know how to program in any programming language, this is very simple to implement. So I wanted to show you some examples. So the first example is the example I've been showing before. It's this human PBMC data set. It has 10,000 cells. It's from the 10x website. So it's a Chromium version 3 protocol. So this is the result that you get from t-SNE after using 30 principal components and a Freeman-Tukey transform, square root based transform. And you have to kind of believe me that these annotations are kind of sensible. But I think they're fairly good. It's the best I can do, at least right now. And you see that you get a pretty nice separation of the main cell types in PBMCs. You can do the same thing with a log transform. So changing from this Freeman-Tukey to a log transform, you see that the result is pretty similar. You get almost the same separation of cell types here. Here's a few cells that I kept the annotations here. So a few of the orange cells that were here before, they're now a little bit closer to red cells. I mean, that's the only big difference, or the only clear difference I see. This shape of the T cell cluster looks a little different. Not sure what that's about. Here there's a few, these are probably doublets or something. They're a little bit different, but like I said, I mean, it does the job. I think again, from a theoretical perspective, the square root transform is better, makes better use of the data, of the signal in the data, but either way works. The second example is from mouse embryonic heart cells, also from the 10x website. Fewer cells, but interesting data set because it's from mouse embryos and developing heart tissue. And here you see a lot more cell types, I think, than in the PBMC data, so I increased the number of principal components to 50. What happens is when you, for example, reduce it to 20, which is clearly too low, you start seeing these cases where cell types that were previously separate, they're now together. So that's obviously bad, so So make sure you include enough principal components. And so, by the way, I actually don't know what all these clusters are, but there's a big cluster, maybe this one here, that's T cells, and I just find that fascinating that there's so many T cells in this hard tissue. So I just wanted to mention that. So for discussion, think of PCA like pancakes, right? So when you're just starting out and you wanted to make some pancakes, you start with a basic pancake recipe, and that makes some pretty good pancakes, you know. So if you just want to have your pancakes, and then this basic recipe is a great starting point. Obviously, there's also fancier versions with nice decorations and jam and fruit, and something where you can barely recognize or see the pancake, but these obviously take more effort, and often in my analyses, I just want to get my pancake and move on, and with whatever other analysis I wanted to do. I don't want to spend my entire day just making pancakes. So what I mean is, there's other PCA variants for single RNA-seq data, and I think they're also very interesting. Two that I wanted to mention are GLM PCA, published this year, where PCA is applied to these residuals that you get after modeling the data, and a little bit similar, I think, is SC transform from last year, where PCA is applied to Pearson residuals. So those are interesting ways of doing PCA for single cell RNA-seq data. They have in common that they are using parametric models, and so it's a very different approach from what I described, because what I described, there's no modeling at all, in whether parametric or non-parametric. It's just you do this transformation and the scaling and the transformation, and that's it essentially. And then I wanted to mention some benchmarking papers, which I think are very, very nice. There's this comparison of different PCA implementations from different Python or from different single cell analysis packages, including Seurat, ScanPy, CellRanger, also scikit-learn, the one I'm using, and how accurate and how fast they are, which I think is very, very useful. Especially, like I said, you wanna have an efficient implementation of PCA. And then there's this other paper which compares 18 different dimensionality reduction methods for single cell RNA-seq data, both linear and nonlinear. So this is a little bit broader, but I think it also includes the GLM PCA, and so that tries to get at how effective these different methods are. But I think it's still fair to say that just PCA by itself is by far the most popular dimensionality reduction method right now, and it's used as it's obviously not, TSNE, for example, is way more popular as a visualization tool. So PCA is not so much a visualization tool, but all the TSNE is basically they are applied to principal component scores. So the PCA is basically the first step of the visualization, and then the TSNE is the second step. And similar with UMAP, because PCA is so good at removing noise from single cell RNA-seq data, it also makes sense to apply UMAP to PC scores, not to the raw data. So these are references. I'll put those in the video description. And thank you for watching, and follow me on Twitter. If you are interested, try my Python single cell RNA-seq package called Monet. For feedback, you can use Twitter or my email. And I wanted to promote this fundraiser that I came across on Twitter recently, by a woman called Savannah Bertrand, and she's asking for money. She said she came out as lesbian to her mother and was abused by her, and is trying to get her own place to live. So I think that's a great way of supporting somebody who can really need the assistance. So if you have some money to spare, consider donating. I think she's still looking for something like $600, $700 to meet her goal, and I think she would appreciate that donation very much. I don't know her personally, but I just think that's a situation where somebody can really use some support. Okay, thanks very much again, and see you next time.